All right, I think we're ready to start again. So hopefully online you can hear me, and if not, put something in the chat and somebody will notice and let me know. So my name is Kate Stumpo. I am the Imaging Business Unit Manager, and I wanted to talk to you today about bringing together some of the proteomics and metabolomics information and how we can bring spatial awareness to that as well. So since we live in a 3D world, whether it's a mammalian, insect, or plant species, the context of cell-to-cell interaction and communications matters. So you've heard about bulk analysis and how we can look at bulk signal, averaging it over the sample in order to have the maximum sensitivity possible. There's been a lot of talk about single cell analysis, so you can understand exactly what's going on within one set system. So I'd like to kind of complete this story by talking about the spatial awareness and how the in situ environment is important for what's going on within a biological system. So MALDI imaging is a type of molecular imaging where we're going to make ion maps of the spatial location of the molecules of interest to us. This is a complementary procedure to some things that I know are already going on here, and there was already some interest from. So doing targeted protein imaging using immunohistochemistry, doing spatial transcriptomics, getting genomics data from fish type techniques. All of this brings together the full transcript to protein and metabolite picture, so you have a full biochemical understanding of what's going on within a system. So what I'd like to show you is how we can do the metabolite, lipid, and protein part of that story all within one platform overall. And then you can overlay multiple types of data sets within our software system and be able to look at everything across that range. So most of the stories that I have for you today are cancer research based, because I knew there were going to be a number of people here from the medical school. And so that's what I put in to focus on. So in this recent publication, metabolic reprogramming during cellular transformation was highlighted. And a number of different techniques were utilized to go through and talk about and build this story. You can see some images up here. Everything that's up here, except for this one that says hexose 6-phosphate, everything is non-MALDI imaging. But that small molecule information was critical to being able to put together the full metabolic picture of what was going on. So the protein side of this was done with cyclic immunofluorescence, which I think is a well-known and commonly used technique. But near the end of my presentation, I'll show you how MALDI imaging can now start to answer these questions and build within this story as well. For the small molecule portion, glycolysis and TCA cycle components are what was critical to use MALDI imaging for, which you can't access in an imaging sense from anything but mass spectrometry. Another example, looking at a multimodal type of imaging within cancer research. So in this one, we again have a competitor technique. This is imaging mass cytometry, where we can look at targeted proteins and their location within a cellular system, looking at one protein at a time. And then in combination with that, histological staining on top, and then a drug target and a labeled drug target of interest in order to give complementary information. So the MALDI imaging is completely necessary to see where drug targets are penetrating within the tissue and what metabolites are resulting from that. And then the protein information can tell you the drug target that's being acted upon. So you have, again, the complete cycle of information available to you. Again, you can do all of this within MALDI imaging. So the way that this works is we take our tissue specimen, put this down on a conductive glass slide surface, and you see a stylized version of that here on the screen. We're going to take a mass spectrum at each XY spatial location that's on that slide. So we're going to generate this average mass spectrum, and then we can go back and interrogate and build the picture at each pixel level. So I can say, show me the green highlighted area, or show me the blue highlighted area, and what is the resultant image and distribution for that. So in this partial kidney sample, you can see ion expression for a variety of different species that are in different locations. We can overlay these to get the nice, pretty pictures that are shown here. And with our Newest add-on, we can get down to 5 microns total for the lowest spatial resolution possible. On your instrument, 10 microns is your lowest spatial resolution possible, but I did want to show just kind of the capabilities that are possible with some of our more recent updates. Okay, so you've seen the instrument a little bit, but I just wanted to have one slide in here to really highlight that you have that quick changeover in the source. So it's literally the click of a software button to go from the ESI modes that Sergio and Erica talked about to going into the MALDI imaging modes. So it's not a complicated breakdown of the instrument or changeover. And then the types of classes of molecules that are accessible, I've got written up here for you. So again, the small molecules, metabolites, and lipids, other endogenous species or small molecules that have been introduced as drug targets. With additional preparation, you can do glycan-based imaging, and I'll show you an example of that. Endogenous peptides, and then the targeted protein workflow, which we call our multi-hiplex IHC workflow. And then you'll hear me talk about some of the instrument components that go along with this as I move through. So for doing an imaging experiment, this can be done from fresh frozen or FFPE tissue. Of those molecular classes, you can get lipids and metabolites from FFPE, but your yield will be lower than from fresh frozen. So that's kind of a new thing that we've been working on in terms of method development to help and assist with recovery of molecules from FFPE-based tissue, because clearly in the clinical world, that's where most things are and how most things are stored. So if you do then have to move on to sectioning and mounting, we provide consumables for that. Then matrix application. So we provide standardized protocols that assist with method development. So you're not starting from scratch. We're giving you kind of a set of tools to begin with so that you can move through and develop exactly what you need. And then the data acquisition and visualization and analysis of the data, we have a full field applications team that can help support. So if you're undertaking a new experiment that might not be as familiar to you, we're happy to lend our user base and our collective knowledge to help with success overall. So again, with the medical school, the cancer focus, and the use of other techniques that you're likely engaging in, I wanted to make sure to mention that we have a plugin for Qpath, which is one of the digital pathology tools that are accessible for pathologists. So if you have a pathologist that goes in and annotates on their H&E staining, that's directly portable into our software skills lab. So those annotations will then come over and you can compare and do all of your MALDI imaging analysis with that already highlighted within your sample sections, which is, I think, good for information content and for capabilities there. And then we have examples and tutorials and whatnot to take you through that if you are embarking on that. The other thing that we have available is a software developers kit, namely an API. So if there are types of classifications, statistical analyses, and things that you want done that are not in our software, we have an API plugin. So if you can code it, you can bring it to the table and you can then output everything within Skills Lab as well. The place where we've started to see some examples of this are looking at labeled and ratio-based experiments. So taking a carbon-13 labeled glucose and looking at how it's affecting the metabolic cycle and then pulling out downstream where it's connecting in a control versus a disease-based tissue. Hopefully we'll incorporate that into our software, but we have to hear the ideas and the use cases from our users in order to have a more expressive module within the software. Okay, so let's go back to some pictures. So that's kind of how you do the experiment and the basics of what's possible with our software and add-ons. So here's a small selection of small molecule analyses. So this was done recently for a pharma customer who was interested in the redox pathways. And so I just pulled out a handful of the molecules that are accessible and that are able to be imaged within that space. In addition, in the small molecule space, we get a lot of questions about the TCA cycle. So here's an example graphically looking at what can be pulled out and visualized consistently from within the TCA cycle. So citrate, isocitrate, aconitate, succinate, fumarate, malate are easily accessible. Some of the other molecules will depend on overall abundance and expression, and if we're able to pull out additional information. But we're at the beginning of being able to do a true metabolic flux analysis within an imaging experiment, and then keeping that spatial location and expression as part of the story. Okay, another way that we like to talk about starting down that data analysis pipeline, and I think this is one of the intimidating places for new users, is, okay, I can generate these images, but what if I don't know what I'm looking for? How do I dig into this and get meaningful information out of all of these gigabytes of data that I've just collected? So in this frozen mouse eye section, you can see a couple of molecules that are expressed, and you can see the resulting images here and how they're in different locations overall within that eye. Then we can do an biologically a priori calculation. So no biological information goes into this, simply an algorithmic way of clustering together the pixels based on the spectra that are generated. So this is what we call a hierarchical clustering technique, in case you are familiar with computer science terms and how that would be done. But the end result of this is I'm looking at pixels that are expressing similar mass spectra. So I have similar cellular phenotypes that are giving me similar mass spectra, and mathematically I'm able to pull that out. So if I zoom into this area and I look at the different cell layers within the retina, and if I count through, I've got green, blue, light blue, purple, yellow, red, yellow, orange. I've got 9 different colors that are showing up here. A mouse retina has 9 different cell layers that are present there. So we're able to pull out that phenotypical information and that there are differences being expressed. I can now go in and say, tell me what's different between the red and the blue region. Tell me what the top 10 ions are for each one that are different. And so now I get actual information that's useful in a biological sense. So let's look at an example of using that segmentation analysis. That's the easy word that we use instead of hierarchical clustering, which I hate saying. So let's look at this within a cancer section. So in this experiment, a number of different prostate cancer sections with different Gleason scores were graded by a pathologist, and the tumor regions were outlined. So in the H&E stain, you can see the black line that outlines the tumor region. If a pathologist was able to tell everything from one stain, that would be ideal. But we know that it's still based on looking at this and making a judgment call. Pathology is a well-developed field that does a fantastic job. But what if we could give more information to those people so that they can make a better choice overall? So after doing a MALDI imaging experiment and doing segmentation analysis, again, with the principles that I talked about in the previous slide, looking at cellular phenotype, I see that within that tumor region, there's more than one color. That suggests that there's more than one cell type or subpopulation of tumor cells that are present in that region. So I have additional information now that I can go back and utilize and see what lipids are important for the grading of those tumor sections and for assigning the Gleason scores. It's just one example of how MALDI imaging can bring additional information to the things that you're already doing. So let's look at an example for N-glycans. So this is a procedure where you have to follow the recipe very, very carefully, but there are kits for this. This is Dr. Rick Drake at the Medical University of South Carolina, who helped to pioneer this and formed a company called Glycopath, and they sell kits for doing glycan analysis specifically from tissue. So after doing the procedure listed there, you get your general mass spectrum with all of the different glycans of interest with putative structures. Remember that with glycans, we can figure out what monomers are present, but we have to use some inference in how they're connected to each other and what's actually going on there. But in this carcinoma sample, a colorectal carcinoma sample, images of the different glycans and where they're present. And we know that mucinous proteins especially give a lot of different expression within cancer systems. been told by a couple of pathologists in the field that they feel like glycans are the next big thing. And so I'm waiting patiently for more and more people to adopt this and bring this into their ideology. Okay. So I told you about a number of different classes of molecules and the importance, if you didn't already know, within cancerous systems. So let me turn to a couple of the instrument features and a few specific workflows that might be useful in the biological questions that you're asking. So your instrument is equipped with TIMS, trapped ion mobility, which was talked about extensively and very thoroughly by Erica and Sergio. But let me show you in an imaging experiment how this is still useful and how it brings additional information content. So if I look at the mass spectrum only, I've got two kind of distinctive peak areas that are present here, which are labeled on the screen there. And then I can pull out the image plus or minus half a Dalton for each of those peak areas, or 0.1 Dalton, whatever I'd like to do. But when I do the TIM separation, and now I have a second dimension of space, and I'm really looking at things based on their shape in space, I get their collisional cross-section out, and now I see there are more hotspots present here in this 2D plot. And so I have so many more images that I can generate, and I can start to look at some of the unique isobaric species that might be present here that I couldn't uncover without that trapped ion mobility separation. All right, so then I'll move into a handful of the workflows that we have that are accessible. And I think ways to utilize your instrument to the best of the possible capabilities. So this is what we call our SpatialOMx Workflow. We're going to combine together MALDI imaging with a full proteomics or metabolomics experiment in order to figure out where should I be looking to do the deepest dive of proteomics possible. That's part of the struggle. That's part of why we do bulk proteomics, is we know that there's going to be a change expressed somewhere. But if we could figure out what cells are causing that change, and we could interrogate just those cells, we have a much more information-rich experiment that's resulting. So what we're going to do here is acquire our MALDI imaging experiment. We'll generate our images. We'll do this segmentation analysis again. And let's say that lime green region is the one where there's something interesting going on based on my genomics analysis, based on my other staining protocols that I'm utilizing combined with my MALDI imaging data. So then I can cut those cells out. So I can do a laser capture microdissection, and I can do a very precise cut, or I can just use a razor and I can cut out as much of that cellular population as possible if I don't need that high of a level of precision. Then I can do a deep dive of proteomics in order to look specifically at just that cellular population and figure out what's going on biochemically there. So let's look at an example. I was a teacher in my former life, so I know to put in an example here to explain what I'm talking about. So I have an H&E stain in the background here, and then I did a lipid imaging experiment, and again, that segmentation analysis has been done. In the dotted line zoomed in area, there are actually 3 colors being expressed within that tumor region. So I'm supposing that there are 3 subtypes of cells that are present. If I cut out each of those populations individually, and then I do a full deep dive LC-MS-MS proteomics experiment, I get the biological activity of each of those subsections, and they're not the same. So I'm gaining more information that I can now tie back to the spatial location overall. So this came out of Ron Heeren's lab, and this was published a handful of years ago. Just last week, he published another experiment very similar to this using our high-plex workflow. So doing a targeted look at where specific proteins are to then pull out the cellular populations of interest. If you heard my woes when I was walking in, my laptop died on the plane on the way here, not a power cord issue. That's why I didn't put another slide in here, but I'll be happy to provide Jen with the references for anything that I've put in the presentation here. So let me get to that next workflow, the MALDI high-plex IHC workflow. It's kind of a mouthful, but really what we're doing is only adding in a couple of additional steps. If you're familiar with IHC at all, it's very similar to a standard protocol. It just has a few tweaks to make it MALDI friendly because MALDI ionizes things. And so we have to make sure to not have certain contaminants there. We're going to do one additional step here. That's a photo cleavage. I'll show you on the next slides what that does and why it's important. And then everything else that I've already talked about here, application of matrix, analysis, and readout. The reason that I'm promoting this workflow to you is there's no conjugation here. The antibodies that you'll use for targeting your proteins of interest are not something that you have to do any additional wet chemistry on in your lab. You can go straight to the IHC protocols. That's in contrast to imaging mass cytometry and to MIBI. This also works on fresh frozen and FFPE. You can look at your entire slide in under an hour if you're going to do a survey of maybe 80 microns or so. If you wanted to drill down and do a higher resolution, like 10 microns, you're going to increase your time for data collection. And that would depend on what resolution you want to go down to. The other advantages are you can multiplex. So there's really no limit to the number of antibodies that you can put onto the tissue because this is a mass spectrum at the end. This is not an optical technique where we have to do a fluorescent readout and we're limited to 6 channels at once. I can read out everything at once. I can do multiple analyses on the same tissue section so that I can do multi-omic or multimodal analyses overall. So let me show you how this works, and then I'll show you a couple examples. So what we're going to do is our MALDI friendly staining protocol. We're going to put down our antibody of interest on the tissue section. The red circle that you see there is a photocleavable moiety, and then there's a unique mass tag that's coming off of the top of this. So what we'll do is we will photocleave that photocleavable moiety, release the peptide mass tag, that's a unique mass that we know, detect, and visualize. And then again, we can multiplex this. This works on the timsTOF Flex, which you have, and then we have easy readout in Skills Lab overall. And then here's what it looks like. So on the left side, I'll explain how this is multimodal in just a moment. On the left side, you see a fluorescent image, and then the same protein done on the same tissue, the MALDI image that results. On this particular one, you see the yellow star that's on there. There's also a fluorescent tag. So you can cross-validate with other methodologies if you want to be sure and have controls built into your experiments overall. And then at the top, you see the name Ambrogen. So we're partnering with Ambrogen. It's another company in the Boston area that actually makes these antibodies, the tags, and does all of the conjugation. So they ship as fully conjugated and ready to go antibodies. So this is the original paper where this came from, came out in 2020. So again, the 2 images that I just showed you, and then a handful of other proteins of interest, and then kind of summarizing pretty much everything I just said. Fast, you can drill down to specific regions of interest if you want high resolution. I've shown you the combined fluorescence and MALDI imaging. I didn't show you doing H&E staining, but you could do that as well at the end. You can wash off the matrix, do H&E staining, and now you've got 3 data sets that are now overlapping that are giving you complementary data. For the people who are interested in the technical side here, here's what the mass spectra look like. So all of the mass tags are between 900 and 1500. So they're easily ionizable species. They do not overlap with matrix peaks, so you have no cross-contamination. So these have been thoughtfully designed overall. And then, as I've said, you can also do multi-omic experiments. So I could do a lipid imaging experiment first, wash off the matrix, do my MALDI-friendly IHC staining, look at my proteins of interest, and then I can take both data sets, put it back into Skills Lab, and I can visualize the lipid, the PI that's listed here, a sterol is the ST, and then 2 proteins of interest that are also being looked at here. And this is from a mouse glioblastoma model. And then this is just a normal mouse brain, again, looking at lipids on the far left side, a few of the targeted proteins, and then some overlaying of things when I get into the full image. So we like brains to show things off because the white matter, gray matter give us especially good contrast and have very different expressions overall. So it's a good model system. So to end with today, and I know I didn't go too terribly long here, but I really wanted to try and tie together the things that you've heard today and show you the starting capabilities. And then it's really your imagination on how you can apply MALDI imaging and what's possible. But the last workflow that I wanted to talk through is quantitation, because this is something that's not talked about often enough in MALDI imaging, and it's something that is absolutely possible. So I'm going to start on the right-hand side here, actually. So the first way that this evolved, and this has been around for a while. This first paper was from 2010. And then the more recent innovations that have come have been in the late 2010s overall. But in this experiment, I have my tissue section of interest and then a control section that's been put down. And then I'm going to spot a dilution series of the drug or compound of interest that I'm looking at. And then I'm going to build a calibration curve. Skills Lab has this quantitation tool built into it. So it will do all of the calculating for you of what the level of expression is for the region of interest that you've highlighted. And this came out of GlaxoSmithKline wanting to do this for some of their drug compounds of interest. And then the further iteration of this is what we call the tissue mimetic model. So on the left-hand side, we made a slurry out of generic tissue. Spiked in the compounds of interest, made a frozen tissue stack out of each of those slurries. You section it, you put it down next to your tissue of interest that you're looking at, build your calibration curve. And then kind of one last thought to go with this, TIMS is also beneficial for quantitation. So we're putting down this one compound of interest, or we're looking for this one compound of interest within a biological matrix. It's very likely there's going to be a mass overlap somewhere. And so by having the TIM separation and being able to have that second dimension of separation, it becomes easier to find my drug molecule and not have to worry about a tissue-related peak that might be present in the biological matrix. So if I do a quick plot of looking at concentration versus normalized intensity, when I have TIMS on, when I'm utilizing that unique instrument feature, I have much better sensitivity overall for my data collection. So I think a picture is worth hopefully several thousand words. So I've given you a slightly shorter end here, but hopefully have opened your mind to the spatial possibilities and capabilities with an instrument that you already have here. So hopefully you can get started and keep Jen busier. Were there any questions from the audience or from online? Absolutely.
Add the MALDI matrix section. You need a sprayer, right?
Yes.
There's no other way to do it.
There is no other consistent way to do it. The sprayer is going to give you reproducible extraction of the compounds, but not too much extraction so that you have translocation of species. So it is a controlled experiment.
And the thing mentioned again, IHC.
Yep.
So I'm not a mass spectrometer. So are you hitting the tissue with a laser?
It's still a MALDI experiment. Yes.
So stay intact.
You're not ablating everything. So your pixel size that you're looking at, you can look at 100 micron size pixel, but maybe you only ablate 10 by 10. So there are options in your experimental design to leave behind tissue. There are also published studies showing that you can go through and ablate the entire piece up to 3 times before you run out of full material present, because it's 10 microns thick. So yes, you do then have a Z dimension that you're dealing with, but it's only 10 microns. So yeah, if it's FFPE, it's only 5 microns typically, maybe 6. So yeah. Any other questions? I don't see any in the chat. Okay, then Jen, absolutely.
It's really difficult when I get inside. I know that you're the world leader for the imaging system. So something like a global. So we want to get some kind of tens of targets from imaging analysis. Then without the problem, some kind of markers. How we can approach for MALDI imaging?
So the question was, how do I get information from a MALDI imaging experiment? If I have a known protein marker, is that what you're saying? So if I'm doing a metabolite experiment. So that's where doing that segmentation analysis is really critical. You have to start by saying which cell types are expressing similar compounds. And so then within the software, you can pit those two areas against each other and say, show me what's upregulated in the control or the disease tissue. Show me what's downregulated in the control or the disease tissue. So it's all within the software. And that's something that we can make sure that Field Apps works with you on. And we can give you a sample data set so that we can talk through that and do a training on that. Yeah.
So how about based on the single cell? So can you grow the cell?
Yeah. You can grow cells on a MALDI imaging slide, and we can provide some guidance there as well. You could also take something like the cell-in-one, and you could spot down single cells, and then you know exactly where they are. So then you know where to image when you go back and want to look at that. So absolutely, it's another great marriage of that single cell theme with MALDI imaging. Any other questions? Nope, nothing on the chat. They were just saying they could hear me earlier.
Oh.
Okay.
Anything for Shorjo?
Anything for Shorjo or for Erica?
Again, we'll share the slides, obviously, and if you have questions, you listen to the recordings and have an aha moment, please feel free to reach out to Griffith, and then he's going to connect us together.
They are really great. So even when I have a problem with the feedback and analyze the data, they always going all of the data and they method everything. If you have any questions, concerns, let us know. We can solve out your issue because we have up here end of the system. We got two class.
And for better or for worse, we're PhDs, so we're curious too and want to dig down and answer the questions.
I sort of have a general question. So one of the things we struggle a little bit with single cell RNA-seq, for example, is the cost prohibits sort of replicating, there are lots of biological replicates. So when you get down to the sensitivity of these sort of single cell analyses, what's the technical? I guess you can't really do technical variability on single cells, but what kind of variability is there at that level?
So that's a very interesting question, and we're learning as we go. We've seen that initially what our understanding was that the size of the cell would actually dictate the protein content, which turns out to be not true. Different size cells will, depending on the treatment, will give you the same amount of protein groups and peptides. So the variability, it honestly has not been judged on large populations yet, because single cell proteomics is a fairly new workflow, and maybe 4 or 5 labs in the country have actually put it down. So there's not enough data to show variability over large sample cohorts. But yeah, that would be something very interesting to assess. Technical variability is minimal if the sample handling and the analysis is carefully accounted for. I mean, CVs of less than 2% variability from batch to batch, but drilling down on population level variability, we don't have enough samples to run nationwide to address that question. Because there are 2 schools of thought in doing single cell proteomics. One group of people who actually work on the technical aspect of formalizing the workflow and the sensitivity and all of that, and that's pioneered by Nicholas Lavov at Northeastern University. And then there is using the system to actually answer biological questions, which is really pioneered by Jenny Van Eyck at Cedars-Sinai. So again, it's very new, and so we'll find out in the next 2 years as to where this all goes.
So you're saying the technical variation in the system is pretty low? Very low.
Yeah, your technical variation batch to batch happens not so much on the system itself, but also on the sample prep side.
Yes.
It's critical. Yeah, it's critical to actually have cell-in-one or something akin to it to minimize the batch to batch variation, because you're dealing with so little amount. If you're not careful about it, you're just gonna be all over the place. You're not gonna be able to make any sense out of it. Okay. Excellent. Anyone else? Go ahead.
Yeah, you provided show us a lot of different software for analysis. Yeah, so far.
For proteomics,
Yeah.
Yeah.
okay. Which software is the best software?
Oh, boy.
No.
I would say, and I'm obligated to say that Phaser is the best as we make it. It depends, honestly. There's some, depending on the workflow, I would say glycoproteomics, DDA Fragger is very good.
Mhm.
For DIA, Spectronaut is a very polished product. Their outputs are very professional, and you can go directly put it on a slide. You can do a multivariate analysis, but it's a paid software. In terms of proteome depth, Phaser is good, and so is DIA-NM. But again, it's freeware, so the interface is a bit rough. And you need to have an expert level understanding of what kind of modifications you want to put. It's not a plug and play solution, unlike Peaks, where it's pretty simple to put everything in. So I mean, everyone has their own favorite flavor. Some people do it in-house, so there's no such thing as best, as it were.
Anything else?
Excellent. Well, thank you once again for your time. Really appreciate hosting.